Thanks, Maggie. Don't know how much I'm going to be moving around, but why not? As Maggie said, I'm a biochemist. I like looking at respiratory proteins and how they evolve. And I infer patterns from large sequence data sets and usually verify them by experiment. Today, I'm really going to be talking a lot more about just the patterns we observe in metagenomes of ANMI and SRB and how we can use that to understand this really cool symbiotic relationship in the deep sea between anaerobic methanotrophic archaea and sulfate-reducing bacteria. So chemosynthesis is really important in deep sea ecosystems where essentially carbon fixation is occurring with the use of reduced electron sources such as methane rather than photosynthesis. And one kind of chemosynthetic reaction that's really important in methane seeps and hydrothermal vents is the anaerobic oxidation of methane. I'm glad someone mentioned facultative mutualism. This is an obligate syntrophic relationship between these archaea, which are and these bacteria, which together form these multicellular aggregates. So the archaea are here in orange or pink, and then the bacteria are in green. So they form these beautiful multicellular aggregates across different types of ecosystems, and they can only exist together because methane oxidation is unfavorable when it happens in anaerobic sediment. And so the only way it can happen is when there's an electron sink, and that is sulfate reduction, which is performed by the bacteria. So what's really interesting is that they occur in different types of ecosystem, making different morphologies, but also this reaction convergently evolved on both the archaeal side and the bacterial side multiple times. There are multiple groups of anaerobic methanotrophic archaea that are capable of performing this methane oxidation. And likewise, there are multiple groups of sulfate-reducing bacteria that are capable of forming this partnership. And all of these I'm going to talk primarily about 4 different clades of this sulfate-reducing bacteria that we've characterized fairly well. These are hot SEEP1, SEEP-SRB2, SEEP-SRB1G, and SEEP-SRB1A. These are all within the phylum Desulfobacteroda and are taxonomically unrelated. So as I mentioned, it was a kind of convergent evolution. What's really interesting is that sometimes some of these archaea and some of these bacteria can form these relationships promiscuously. So they can form relationships and form these multicellular aggregates with different groups of bacteria and different groups of archaea. So what I'm really interested in is asking, so what is important for the, this is me showing. We looked at these in all of these different places in the world. You can see the exact same multicellular aggregates. What I'm really interested in asking is, how do they form these aggregates? What defines the symbioses, both in terms of the physiological nature of them and the physical components of these? And by understanding this, how can we also understand how a free-living sulfate-reducing bacteria might become this mutualistic partner? So these are the questions that I'm interested in asking. And so how do we do that? So I think of this as kind of the way that you look at, just like you look at the evolution of different anatomical components using fossils, you can do the same thing by looking at physiological pathways and physiological traits as things that evolve in organisms. If you understand the evolutionary relationships between the microorganisms and you understand the physiological traits that make them up, you can see how they evolve. And so that's what we'll be doing. And so our data set for doing this was basically, so in order to understand the evolutionary relationships, we used the Genome Taxonomy database and used basically the entire phylum of bacteria of which these organisms are a part. That's about 550 odd Desulfobacteroda genomes. And then we curated a whole bunch of these syntrophic organisms across different ecosystems and forming partnerships with different types of ANMI. So this involved a fair amount of metagenomic sequencing, both from us and from other groups, as well as we are able to make these partnership associations based on fluorescence in situ microscopy, in situ hybridization microscopy, and using single aggregate sequencing, where we basically take these multiple these multicellular aggregates and sequence them individually to know which genome is associated with which partnership. So using all of this, we basically have context for where they're from, who the partners are, and what their taxonomic context is. So once you do that, and then you also develop this physiological model for what this mutualism actually involves, you can then look at the physiological traits like these anatomical components that I was talking about by basically looking at the presence and absence of genes in the entire evolutionary neighborhood of Desulfobacterota. And you can look at phylogenetic trees of each of these corresponding proteins that form a component and see how it evolves. Basically, it's a little bit like gene and species reconciliation, but in a slightly more manual way, where you're basically saying, if this is an example of an inner membrane component, you can see that it's largely absent in Desulfobacterota, except in the one order of metal-reducing organisms like Desulframonidalis. And then you can also see when you generate a tree using all the genes from publicly available data sets that it is most closely related to a taxonomically distant group. And you can infer that it's a horizontal gene transfer. So in order to do this, then let's make the physiological model first. What does ANMI-SRB syntrophy involve? And so this is my description of what the biofilm looks like when you zoom in. You basically have an extracellular matrix with polysaccharides, much like Pseudomonas and so on and so forth. And you have structural components such as type 4 pili and adhesins that make up all of these components in a biofilm. In addition to that, you have these essentially what drives ANMI-SRB partnership is direct transfer of electrons from the anaerobic methanotrophic archaea to the sulfate-reducing bacteria. So as I said, the only way this works is if the electrons that come from methane oxidation go into the bacteria. And how that happens is through a conductive biofilm that has these heme-containing nanowires that can move electrons. So we inferred what components are important by basically using a lens of comparative biology. So these are multicellular aggregates and they're conductive biofilms. So what is important for Geobacter sulfur-reducens, which is an extracellular electron transferring microorganism that forms conductive biofilms? We expect similarities in what's found in ANMI-SRB. Likewise, we use Myxococcus xanthus, which is a multicellular fruiting body, and says what provides that kind of interspecies communication and formation. And then we use the components that are similar to that and see if it's found in ANMI and SRB. Ditto for Pseudomonas, which forms another biofilm as well. So by using what is found in similar organisms and comparing it, we can identify what's important for the physiology of Okay, next slide does not want to load. Yeah. There you go. All right. So first part of this, as I mentioned, the only way this can happen is if electrons can be transferred straight from ANMI to SRB. So the proteins that are involved in this are basically these extracellular electron transfer containing heme wires. Those are the polymers of these proteins that have hemes in them. That's like OMCX and OMCS and so on. Now, the electrons that are outside the cell need to get inside using proteins in the outer membrane. And then through the periplasm, they go into the inner membrane and the respiratory pathway that ultimately donates electrons to sulfate reduction. So everything that's in the inner membrane and below and in the cytoplasm is common to all sulfate-reducing bacteria. But you needed all of these components that exist in the outer membrane and extracellular space to form an ANMI-SRB partnership. So where do those come from? And this is kind of our analysis based on all the phylogenomics and comparative genomics. We can see that this electron transport chain is different in each clade of syntrophic SRB, and that we can also show that in each of these different clades, different things are adapted. In the three clades, CPSRB 1A, 1G, and HOTCEP1, All that's adapted is the extracellular space in the outer membrane and some of the periplasmic cytochromes. In CPSRB2, what you also needed to change are the inner membrane proteins. So basically what it's telling you is that they're taking different evolutionary trajectories to their final goal of forming a syntrophic partnership. And this is the biofilm formation interspecies communication component. As I mentioned, there must be polysaccharides that form up the matrix of the biofilm, as well as type 4 pili and other types of structural components. So all of these need to be present in these, and they are present in all 4 syntrophic SRB, but they're slightly different. So you're able to tell that the CPSRB1A and 1G have different types of components for the polysaccharide biosynthesis pathway. So all of these things that we see in genomes are also corroborated by transcriptomic and proteomic data that we have either from our lab or from other labs. So we're using both the lens of existing physiologic experiments as well as using this evolutionary analysis to say, if evolution preserves something, it must be physiologically important, and that is supported by the omics data that you see as well. And so another really interesting thing that we see is there's these highly specific cohesin and dockerin complexes that were previously found in ANMI are also found in SRB. So here are these, there are some components that are very similar to other types of biofilm formation pathways and other types of conductive biofilms. And then there are some additional ones that are unique to this syntrophic partnership. So we're basically using that to define this physiological model. The next is essentially, this is looking at how these genes were acquired. How many of these were present in the nearest evolutionary ancestor? Those are marked as vertical gene transfers. And then what genes that are important for the physiology are gained through horizontal gene transfer, and what genes are lost. So this allows us to get an idea for what kind of components were lost in the formation of the partnership and what was gained and what was just adapted. What we see typically is that things that are part of the extracellular electron transfer pathway, many of them were vertically transferred and therefore were already present in the ancestor of this syntrophic organism. So that tells you that the ancestor was already capable of extracellular electron transfer, and in many cases. So that suggests that some of these were perhaps metal cycling organisms like Geobacter that exist in the environment, and they found an ANMI partner and then went towards the mutualism. But that may not be true for all of them. One case at least looks like it's a potential horizontal gene transfer. And in all of these cases, FDH and so basically the first two components are extracellular electron transfer related. FDH and HYD are formate dehydrogenase and hydrogenases. These are components that are lost because they no longer need to use diffusible things like hydrogen and formate. And then some of these clades you can see are also adapting their inner membrane protein. But the most important thing here is that almost a lot of the biofilm formation and intercellular communication stuff are horizontally acquired. It suggests that that drove a lot of the adaptation towards the syntrophy, whereas they already had the respiratory pathways that were needed to do this. And the other interesting thing that we were looking for is there any partnership specificity? Why do they form so many different types of partnerships? And is the speciation that we're seeing within clades of these syntrophic partners driven at all by their coevolution with archaea? And one interesting example that we see is within cobalamin biosynthesis pathway. What we see is that within the same genus of organism, some species that partner one type of archaea have cobalamin biosynthesis, while the other partner has lost that cobalamin biosynthesis pathway, showing that there is specialization within the metabolism. So we're looking forward to following up on all of that. But yeah, the major conclusion is that it looks for like for a lot of this, the EET pathways were already present, and they needed to gain the biofilm formation as well as interspecies communication, which gives us some idea for how these free-living SRB must have evolved to a syntrophic partnership, possibly metal-reducing organisms finding their favorite archaea and forming a multicellular aggregate. So our future directions are, as I mentioned, I like looking at some of these experimentally and general heme-containing proteins and respiratory proteins. So we heterologously expressed some of that extracellular electron transfer machinery in Geobacter, and we're able to show that it is electrically active. More characterization is going on, and we'll look at these later. Thanks to everyone, funding sources, and the organizers as well.